Well, thank you, Johan, for the introduction. And I'm delighted to be here in Panama and to present our research. Thank you, Dr. Rao, Dr. Britton, and the organizers for the invitation. I'm going to present about some of the our recent discoveries on genome damage and DNA repair deficiencies, and their implications in motor neuron disease. Before I begin my talk, I would like to mention that the major focus of our research in our laboratory is on three neurodegenerative diseases: motor neuron disease, ALS, Parkinson's disease, and stroke. But today I am going to focus on one of the joint projects we initiated 2 to 3 years back with Dr. Rao in INDICASAT with the support of Dr. Arturo Melo, and two graduate students from Panama, Erica Guerrero and Valmarini Vasquez, has been working on this project for the last 2 years, and they have contributed immensely to this project. So as you know, there are more than 200 different types of neurodegenerative diseases identified in humans, and most of them are sporadic, like without any known causes, while 5 to 10% could be inherited with specific mutations in a specific protein, in a key protein. Now, one unique feature among all these neurodegenerative diseases is they have common etiologies. Although the region of the brain or the spinal cord affected is different, they seem to have similar etiologies or common etiological factors, which could be segregated into three major groups. One is oxidative stress and metal toxicity. Second is protein misfolding. And in each of these diseases, one or two key proteins seems to be aggregating or misfolding. For example, amyloid beta or tau in Alzheimer's, alpha-synuclein in Parkinson's disease, which have been the most studied misfolded proteins along with prions. And the more recent ones are a class of proteins called RNA-DNA binding proteins. These are nucleic acid binding proteins called TDP43 and FUS, and they have been implicated in motor neuron diseases, ALS and Parkinson's disease and so on, and that is one of the focus of my talk today. And the third common etiological factor among all these diseases is accumulation of various types of DNA damage. And what is not known, however, is the relative contribution of these factors towards the phenotype, as well as which is the primary or which is the secondary contribution to the phenotype. And I should also mention here that despite several decades of intense research, we do not have a complete cure for any of these diseases. The available therapies can only delay the symptoms, but they do not stall the neurodegeneration. So, as my focus of my talk is on ALS, I want to give a brief introduction about ALS. ALS is amyotrophic lateral sclerosis, which is popularly known as Lou Gehrig disease in US and I'm sure in Panama as well. And this is characterized by progressive degeneration of upper and lower motor neurons in spinal cord, and that leads to the motor symptoms, as well as patients are unable to breathe, and they will have to be put on ventilator. And this is one of the although this is a rare form of disease compared to Alzheimer's and Parkinson's, this is one of the most lethal or severe form of neurodegenerative diseases, because the patients die within 2 to 3 years of diagnosis. And as I mentioned, the major our recent discovery, our recent studies are focused on genome damage and implications of genome damage deficiencies in neurodegenerative diseases. The brain is one of the most susceptible organs for genome damage, because brain consumes most of the oxygen we breathe, and that leads to reactive oxygen species, byproducts, and oxidative stress. And even otherwise, DNA is inherently susceptible to DNA damage, both endogenous as well as exogenous, as we are exposed to radiation and other drugs for diagnostics as well as therapy. And these agents cause multiple types of genome damage. They could be oxidizer bases, DNA cross-links, single-strand breaks, and double-strand breaks. However, all these damages are efficiently repaired in healthy cells, and there are multiple primary and backup pathways to repair this DNA damage. For example, oxidizer bases are repaired by base excision repair pathway, which also shares the machinery with single-strand break damage repair process. And the most lethal form of the damage, double strand breaks, are repaired by 3 repair pathways, which I am going to show in the next slide. And I would also like to mention that in a healthy mammalian cell, about 10 to the power of 6 base damages occur in a cell every day. And this looks like a huge number. In fact, this discovery got Nobel Prize for Thomas Lindahl this year for chemistry. Although this number looks a huge number, we think this is still an underestimate because you cannot precisely calculate the number of damage because they are continuously being repaired. So what it underscores is that this huge number of damage is efficiently repaired if the cell has to survive. So this underscores the importance of DNA repair pathways. And for double strand breaks, which are the most lethal form of the damage, in mammalian cells, there are 3 different repair pathways. One is homologous recombination, which utilizes the sister chromatid of the duplicator chromosome as a template to repair the damage, and that occurs only in cycling cells. And the predominant form of double strand break repair pathway is called non-homologous end joining, which utilizes a key protein called Ku. And the third, more recently discovered repair pathway is alternative end joining, which co-opts the single strand break repair machinery like PARP1 to repair the double strand break repair pathway. And coming back to the oxidizer-based damage, we have been working on characterizing the complexities of oxidizer-based damage repair in higher organisms like mammalian cells and humans. And in the last few years, we identified several key features. One of them is in human and mammalian cells, for endogenous oxidizer-based damage, this is carried out by preformed repair complexes rather than recruitment of individual proteins. So there are large preformed complexes containing all the required components of the repair machinery which carries out the damage. And this dynamic complex is regulated by disordered regions, elongated disordered regions in the early DNA repair proteins. And the other key feature in mammalian BER is presence of or participation of many non-canonical repair proteins, like the proteins which were not previously known to participate in DNA repair were shown to be directly or indirectly involved in DNA damage repair. And one such class of proteins is RNA binding proteins. Like here we showed that HNRNPU participates in both oxidizer-based repair and in coordination of oxidizer-based repair with double strand break repair when there is a cluster damage. And during the same time, 2 other RNA binding proteins like TDP43 and FUS were implicated in motor neuron disease ALS by the group of Virginia Lee in 2006. And a unique feature of TDP43 proteinopathy in ALS is it is predominantly nuclear in postmitotic neurons, but in ALS and in other neurodegenerative diseases where the diseases which show TDP43 pathology, the protein migrates to cytoplasm and gets sequestered there. And it also aggregates like any other amyloidogenic protein. And TDP43 being an RNA binding protein, the most of the studies since its discovery was focused on its RNA binding functions or loss of RNA binding in RNA metabolism because of its functional loss. But TDP43 also binds to DNA, however its role in DNA repair was not characterized. And in 2010, Paul Taylor from St. Jude's identified a DNA repair protein Ku, as I mentioned, alluded to earlier, a DNA NHEJ key NHEJ protein Ku in TDP43 immunocomplex. So this in fact prompted us to look into role of TDP43 in genome damage repair and its implications in ALS, and that's when we and Dr. Rao initiated this project. So I should also mention here that ever since TDP43 was implicated in ALS in 2006, several subsequent studies have shown that about 40% of all neurodegenerative diseases, which include the common Alzheimer's and Parkinson's disease, have some kind of TDP43 pathology. So following up on Paul Taylor's discovery of presence of Ku in TDP43 immunocomplex, here we show that TDP43 stably associates with many double-strand break repair proteins, including Ku, DNAPKCS, XRCC4 ligase 4, which are the the major ligases involved in double-strand break repair, double-strand break damage sealing. And interestingly, these damages, these interactions enhance significantly if you induce DNA damage by radiation. And this is done by an assay called proximity ligation assay, which is specific for protein-protein interactions in cell. And we carried out these experiments in multiple cell lines, including SHSY cells, as well as human iPSC-derived motor neurons, or stem cell-derived motor neurons. And this was also confirmed by traditional co-immunoprecipitation assays, where you can see the specific association of TDP43 with double-strand break damage-related ligases like XRCC4 ligase 4, but not with XRCC1 and ligase 3, which are involved in single-strand break repair or oxidizer base repair. And here we show that TDP43 goes to the site of DNA double-strand breaks. If you induce double-strand break in a neuronal cell, within 1 to 3 minutes, TDP43 goes to the site of double-strand breaks. And this is again done by proximity ligation assay with TDP43's association with gamma-2X, which is the marker of double-strand breaks. You can see that when you induce double-strand break by radiation or a radiomimetic bleomycin, TDP43 associates with gamma-2X within 5 minutes. And this was confirmed by another method called damaged DNA immunoprecipitation, where we specifically tag the double-strand breaks with biotin-related UTP in a tunnel sort of labeling, and then subject the DNA to chromatin immunoprecipitation, chip pre-chip analysis. And here you can see that both biotin chip and TDP43 chip amplify same genome sequences that suggest that TDP43 is enriched close to the site of double-strand breaks. And then we down-regulated TDP43 using lentiviral shRNA in motor neurons, and this led to significant accumulation of DNA double-strand breaks, as we measured by a combination of alkaline and neutral comet assay. As you know, alkaline comet assay represents both single and double-strand breaks, while neutral comet assay represents exclusive double-strand breaks. So similar level of strand breaks in both alkaline and neutral comet assay here suggest that most of the strand breaks are double-strand breaks. And this is again consistent with appearance of double-strand break markers like gamma-2X and phospho-ATM, which is a master regulator of double-strand break response. And again, the presence of damage in TDP43 knockout cells was confirmed by long amplicon PCR analysis, where we isolate a large fragment of DNA and amplify it by PCR. So if there are strand breaks, you expect to have reduced amplification, and that's exactly what we observed in TDP43 down-regulated cells. And then we knocked out TDP43 using recently developed CRISPR-Cas9 method, and TDP43 is an essential protein. So the constitutive knockout using CRISPR, the cells died within 2 to 3 days. So we did a conditional knockout, and with a doxycycline treatment, you can see the gradual decrease in TDP43. And interestingly, this is consistent with our previous data. This led to significant accumulation of gamma-2X, phospho-ATM, and more interestingly, 2 other DNA double-strand break response proteins like 53BP1 and MDC1 were down-regulated, were depleted. So although I am not showing the data here, so our more preliminary analysis suggests that this is due to regulation of 2 microRNAs by TDP43, which in turn regulate these 2 proteins. So it appears that the down-regulation of these DNA DDR proteins is one of the reasons why TDP43 knockout cells accumulate significant level of double-strand breaks. And again, with collaboration of Professor Bordog in UTMB, who is also here, who is presenting in the evening session, we showed that TDP43 knockout cells, CRISPR knockout cells, also overexpress NF-kappa B, 2 NF-kappa B fragments, and it appears that NF-kappa B is also activated as their DNA binding increases. Although this is a preliminary study, our data shows that this NF-kappa B activation requires ATM activation in TDP43 knockout cells. So if you block ATM, then NF-kappa B activation is reduced. So this suggests that one of the reasons for this NF-kappa B activation is DNA damage. So, but this is still in the initial stages, and we are going to investigate it further to see what is the role of neuroinflammation and DDR response in contributing to neuronal cell death in TDP43 knockout cells. And consistent with the data, with the accumulation of DNA double-strand breaks, TDP43 down-regulated cells are sensitized to DNA damaging agents like radiation and various other drugs. And the one other dimension to these studies is, as I mentioned, TDP43 is predominantly nuclear in healthy neurons, and it accumulates in the cytoplasm. More than 50 to 60% of proteins at diagnosis is in the cytoplasm, sequestered in cytoplasm in ALS. So the question is, this accumulation of DNA double-strand breaks is due to its loss of function in the nucleus or depletion of nuclear protein, or because it aggregates, whether it's an indirect effect of its aggregation. So to dissect this, what we did was TDP43, because it also migrates between nucleus and cytoplasm, it has both nuclear localization and nuclear export signals which have been identified. And we specifically mutated nuclear localization signal and nuclear export signal so that you can express the protein in the cytoplasm while it's depleted in the nucleus or vice versa. So you can see here that its nuclear specific depletion leads to accumulation of double-strand breaks as measured by Comet assay, while its cytoplasm specific depletion was similar to wild type cells. So this suggests that nuclear TDP43 is required for efficient DNA double-strand break repair response in neuronal cells. Okay, thank you. So, and consistent with this, we also confirm this with DNA damage repair marker analysis, where the nuclear specific depletion of TDP43 led to accumulation of gamma 2X in the nucleus, similar to TDP43 downregulated cells. So these data suggest that nuclear TDP43 is required for efficient DNA double-strand break repair response in postmitotic neurons, and its pathology could lead to accumulation of DNA double-strand breaks, and that should contribute to neurodegeneration. And following up on this data that nuclear TDP43 is required for double-strand break repair response, so we are generating a mouse model, again supported by Dr. Arturo Mello. So we are specifically deleting the NLS signal, which is in a short exon 3, which is about 20 amino acids, and we are using again CRISPR-Cas9. We are generating an inducible mouse model where this exon is deleted with a flox cassette, and right now we have this mouse model and we are characterizing this for the phenotype. So we do not have an appropriate mouse model for TDP43 because the two major pathological phenomenon in ALS is one is gain of toxicity and other is loss of function. Most of the knockout or knockdown models only show loss of function, while overexpression models show gain of toxicity. And we have many transgenic models, but which express human proteins, but many of them transiently show motor phenotype, but in the long run they fail to sustain the motor neurodegenerative phenotype. So we are very hopeful on this mouse model, and we have just received this from the company. This is custom generated, and we are characterizing the phenotype of this mouse model. And we then examined the TDP43 pathology and DNA damage, accumulation of DNA damage in human postmortem tissue affected by ALS. And you can see here that there is a typical nuclear clearance of TDP43 in both motor cortex as well as spinal cord of ALS affected human brain tissue, which is typical of ALS as observed by Virginia Lee and others. And it also has significant accumulation of aggregated proteins, which is again typical of any neurodegenerative diseases. But what is interesting is ALS affected TDP43 ALS patient cortex as well as spinal cords accumulate significant level of DNA damage as measured by gamma 2X staining as well as tunnel assay, and this is the strand break damage. And again, there are several mutations which are implicated in inherited ALS, and 3 of them are most studied ones, and we are working on these 3 mutations. And these mutations, when you express in motor neurons and differentiate the iPSC-derived cells into motor neurons, they lead to nuclear clearance as observed in ALS patients, and that leads to significant DNA damage accumulation. And again, many of the etiological factors like pro-oxidant metals, even diesel exhaust and so on, they also promote nuclear clearance of TDP43. So in summary, in conclusion about the TDP43 story, we propose that etiological factors implicated in ALS as well as TDP43 mutations promote nuclear clearance, and that leads to deficient DNA double-strand break repair and repair response in motor neurons, and the resulting persistent accumulation of DNA damage leads to DNA damage signaling, including ATM and NF-kappa B activation, and that contributes to neurodegeneration. So we are, we are trying to understand whether we can target or modulate DNA damage response signaling to protect neurons from DNA damage induced cell death, and at the same time modulate, activate DNA repair so that you can prevent the neuronal cell death. And another aspect we are looking into is like, what is the role of RNA binding proteins in DNA repair? Few years back, Leonard Muke and colleagues showed small non-coding RNAs like 19 to 21 base pair RNAs are present at the site of DNA damage. We do not know what these RNAs do. It could be transcriptional role, or it could be like a structural role. But we are wondering whether the presence of these RNA binding proteins, TDP43, we showed that HNRNPU goes to DNA damage sites, whether it is along with these RNAs, whether these proteins act as a scaffold or glue to stabilize large DNA repair complexes. For double strand breaks, it could involve like 2 to 3 dozen proteins. And finally, I would like to mention another project which we have initiated in the last 1 year on Parkinson's disease. Again, it involves one of the graduate students from Panama, Valmerny Vasquez. So that's the reason I would like to briefly mention about this project. So we have some very interesting data on mitochondrial toxicity in Parkinson's disease. So Parkinson's disease, as you know, is primarily associated with alpha-synuclein pathology, and many investigations have shown that even in mouse models or in human tissue, synuclein goes to mitochondria. So misfolded proteins have been identified in mitochondria at a certain level. So what we observed was the targeting of synuclein to mitochondria in dopaminergic neurons leads to selective degradation of TOM40, which is a outer membrane proteins. Mitochondrial membrane has outer membrane proteins TOM40, TOM20, and inner membrane protein TIMs, and these regulate the mitochondrial pores, which to regulate import of nuclear coded proteins into mitochondria. And interestingly, overexpression of synuclein or targeting synuclein to mitochondria leads to selective degradation of TOM40, but TOM20 and other mitochondrial proteins are not affected. And this, our data shows that this degradation of TOM40 leads to deficiency of nuclear coded proteins in mitochondria. Many of these proteins are trapped on the outer mitochondrial membrane, and this leads to deficiency of DNA repair proteins as well as antioxidant enzymes in the mitochondria. You can see that, and this also correlates with the aggregation of synuclein, and many of these Parkinson's etiological factors like pro-oxidants and 6-hydroxydopamine, they promote degradation of TOM40. And we also know that this involves Parkin-mediated ubiquitination pathway, and we believe that this is the misfolded protein response in mitochondria. So what we are trying to, this is a very interesting topic, and again in collaboration with Steve Bordeaux, we show that this leads to multiple mitochondrial pathology parameters like decrease in membrane potential and so on. But what we are trying to focus on is, and we are also looking into many Parkinson's patient-derived cell lines, iPSC cell lines, where the synuclein gene is triplicated, where the synuclein is overexpressed, and we observe the similar phenomenon. And what we are trying to focus is how much this contributes to Parkinson's disease neurodegeneration, and where we can target this. Synuclein is not an essential protein in adult neurons, so whether we can prevent synuclein going into mitochondria, or whether we can prevent TOM40 degradation or overexpress or deliver TOM40 by adenoviral means. And we are currently testing multiple therapeutic approaches to prevent this mitochondrial toxicity. And finally, our TDP43 story as well as my Parkinson's disease story suggests that DNA damage and accumulation of DNA damage, repair deficiencies, and defective DNA damage response could play a key role in neurodegenerative diseases. While we always knew that DNA damage accumulates in neurodegenerative diseases, in motor neurons as well as affected other brain neurons, but it was never considered as one of the therapeutic targets, but because researchers always believed that it could be a consequence. So many of these studies, we and others have shown that this could play a key role at some stage in dictating the neuronal cell death, and we believe that this should be one of the parameters in the therapeutic regime. And the overall goal of research in our laboratory is, again, how the etiological factors implicated in neurodegenerative diseases not only induce DNA damage, but also lead to inhibition of DNA repair or deficiency in DNA repair, as occurs in TDP43 case. And this leads to imbalance in DNA damage and repair, and that will lead to several signaling parameters. And many of these factors could, as you very well know, this could initiate several years before you see the symptoms. Like in a classical case of ALS in Guam, people know that this is in people in Guam, they used to consume BMA and unnatural amino acids. And even after people moved from Guam Islands to mainland US, after 30 years, they used to get ALS. So this suggests that many of the disease process would have started 20, 30 years before symptoms appear. So it's almost like futile to go back to see what caused the disease. It could be a complex etiology. But we know that the imbalance in DNA damage repair that directly correlates with the neuronal cell death. And we think, at this stage at least, we can intervene so that we can slow down the neuronal cell and explore avenues to activate repair so that cells survive longer. And finally, I would like to mention people who have contributed to these studies. As I mentioned, Erica Guerrero and Valmarini Vasquez, the two graduate students of Pranama, contributed significantly, and we have other seniors in the lab, Haibo Wang, Dr. Sharma, Joy Mitra, Prakash, Suganya, and others have contributed also to these studies. And I would like to mention Jagannath Rao, with whom I did PhD, and we are continuously collaborating and publishing multiple papers. And this is also a joint project with him. I would like to mention Professor Mitra, with whom I did postdoc in UTMB. And many of the DNA repair aspects I learned while working with this lab. And we have been collaborating with Professor Bordeaux to study the DNA damage repair signaling, particularly inflammation. And many other collaborators, including Professor Garotto, who provided many of the brain tissue utilized in the study, and Dr. George Perry in San Antonio, Alan Tompkinson, who provided ligases, which we utilized in the study. And funding from various organizations. The Mello Foundation, with the support of Professor Arturo Mello, we initiated this project 3 years back, and that led to many other grants, including NIH and MDA grants in the last few years. Thank you so much for listening, and I will be glad to take any questions.
Yes, thank you very much. So I have two questions. The first one I want to ask is that TDP43 causes different diseases, but why? Like, why instead of one disease, it causes different neurodegenerative diseases? Like, how is the action on it?
No, TDP43 primarily causes, motor neuron disease. Its mutations have been primarily involved in motor neuron disease like ALS, while in other Alzheimer's and other diseases, although initially you don't see TDP43 pathology, in a severe form of disease, you see TDP43 pathology, and that could contribute to certain level. And we do not think that TDP43 is the primary factor in those diseases. And it is same with, in amyloid toxicity or tau and these diseases. So amyloid beta could be primary in Alzheimer's disease, but in some cases you also see synuclein pathology. And this is, when the patients live longer, I think they develop more pathologies. But TDP43 pathology is manifested mainly in motor neurons.
So my other question involves is, for CRISPR treatment, how far-reaching it is, and is it even possible?
Sorry?
For a treatment using CRISPR or Cas9?
Treatment?
Yes, for ALS.
No, we have not been using, exploring CRISPR for treatment. So what we are doing is, developing a mouse model with CRISPR, not for gene delivery. So it's like, modulating the gene knockout and looking at the phenotype as a mouse model, not for therapeutic aspect. And Steve?
Dr. Hegde, thank you for this very nice presentation, and I realized something. We discussed this several occasions, but it just popped into my mind. The TDP43 is an RNA binding protein. So do you think that this protein is carrying RNA template to the nucleus to repair the double-stranded DNA, to give a strand to start DNA double-strand break repair?
Yeah, that's a very interesting question. So as you know, in the last couple of years, there are few papers which show that, for double strand break damage, most of the double strand break repair by NHEJ, if it is like oxidative stress induces secondary damage, NHEJ should be error prone, because only the error-free repair is homologous recombination, so which cannot happen in post-mitotic cells. And most two other repair pathways like alternative and joining NHEJ should be error prone. So if these are the error prone repair which is occurring in neurons, so cells should not survive. So there should be some other ways cells somehow repair them in an error-free manner. And one of the possibilities is utilizing RNA as a template, because now you see several RNAs like non-coding RNAs and multiple RNAs, and it looks like the whole genome has RNA, so some RNA for it. And it is possible that cells utilize those RNAs as a template to repair double strand breaks. And one of our collaborators, both Dr. Bodog and my collaborator Dr. Hajra, shows that there is some reverse transcriptase activity at the double strand break response site, and if you block that reverse transcriptase activity, the repair is inhibited. It looks like RNA could act as a template. And I did not mention, we have some very recent data which show that in TDP43 knockout cells, they accumulate many R loops, which are the blocked transcription sites. The RNA DNA hybrids, transcription is like replicative stress. At transcription stress, it forms RNA DNA hybrids, and TDP43 knockout cells accumulate many of these sites. And it's definitely possible that TDP43 transports these RNAs or stabilize these RNA templates at the double strand break. But that will be very interesting to see.
Two questions. First, I would like to say a very impressive tour de force. What's driving TDP43 out of the nucleus when it's used when it's needed in the nucleus? What is driving the movement of TDP43 out of the nucleus at the time when you would think it's needed there?
TDP43, in a healthy neurons, about 90% of TDP43 we always see in the nucleus, especially if we do microscopy, you see mostly it's in the nucleus. But always about 5 to 10% is in the cytoplasm. And studies have implicated that TDP43 transports messenger RNA to the cytoplasm, and this is regulated by some phosphorylation events. There are multiple phosphorylation sites. But in disease condition, studies have shown that this is hyperphosphorylated. So protein needs to be dephosphorylated to get back into the nucleus, but this accumulated protein is hyperphosphorylated. This is like saturated phosphorylation sites. So that presumably prevents its getting back into the nucleus. And many of the mutations, of course, they have more cytoplasmic accumulation, although it may not be directly involved in phosphorylation, but it may be structurally it may accumulate those hyperphosphorylation events.
The second question is a speculative one. If it goes into the cytoplasm, does it go into mitochondria as well? So we know some RNA binding proteins, interestingly telomerase, do that.
Yeah. So no papers have been published, but we know that that goes to mitochondria. In fact, I was reviewing a grant on TDP43 in mitochondria, and we do not know at what level, but some protein goes to mitochondria. And as you know now, initially people thought there are very few proteins in mitochondria, but now it looks like almost all proteins go to mitochondria. So they may have some role in mitochondria. Thank you so much. Thank you.